Hello everyone. Now let's talk about muscle physiology part 3. In the first part we talked about the structure of the muscle and cross-bridge cycle. In the second part we talked about the structure of neuromuscular junction and excitation contraction coupling, and also what kind of factors influencing the muscle contraction force. We will continue on the contraction force in part 3, and then we would also talk about different types of muscle contraction, muscle receptors. Then at the end we will connect to how do we study muscle physiology. We start with muscle fatigue. We said that muscle fatigue is another factor that would influence the muscle contraction force. So what is muscle fatigue and how does it affect the muscle contraction? You would study the details of muscle fatigue in exercise physiology in level 2, but we would have a brief introduction to muscle fatigue now. Broadly speaking, muscle fatigue is a decline in muscle's ability to maintain a constant force of contraction during repetitive stimulation. So when the contraction continues being repeated, muscle can no longer maintain the same level of contractile force. Then we say that the muscle is being fatigued. So muscle fatigue is also a protective mechanism because it protects the muscles from overusing, from excessive using, and from damage, from injury, because the force is reduced, then some of the muscle now is at rest. So that is muscle fatigue. Muscle fatigue can be divided into central and peripheral fatigue. Sometimes it could be the psychological, sometimes it could be the physical. So muscle fatigues have different location. And the muscle fatigue, how the muscle fatigue would happen dependent on the type of stimulation and also dependent on the type of muscle fibers. When we have slow muscle contraction, the muscle produces lower force and slower relaxation. Then the muscle is also slow in fatigue. So that is later, more difficult to be fatigued. So here is an illustration of the top diagram shows how the stimuli is continuing and with each single twitch with a constant force. And then the middle panel showed if fatigue occurs, if the frequency is increased, then the muscle can no longer maintain the same level of contraction force. So the contraction force is declined. That's what we say that the muscle is fatigued. In the lower panel, it shows when a tetanus occurred, when the fatigue is the slower in relaxation as well. So when after a relaxing resting period, then this tetanus can no longer be maintained for that long. Fatigue can occur earlier, and with the fatigue, it shows that it's not only the reduce in the muscle contraction force, but it's also the slower in relaxation. Now, we would like to talk about the types of muscle contraction. First type is isometric contraction. If you do a hand gripping, then the muscle that you're using is not shortening with your arm. The arm that produces force for the muscle to contract, but the muscle itself does not shorten. When the muscle is not shortened, then no mechanical work is done. of course, biological work is done by breaking down the chemical energy, but no mechanical work is done because you're not moving an object from one distance to the other. And in this case, if the load is bigger than the tension, what do we mean by that? That is, when we have the isometric contraction, the force that it can generate is not as big as the load. So the load is 20 grams, but if the muscle does not generate more than 20 grams, then the muscle cannot lift the load. And in the middle one is if the contraction force is bigger than the load, then the muscle can shorten, the load can be lifted, and that is the case with isotonic contraction. So if we go back and look at the isotonic contraction, during isotonic contraction, the muscle produces constant force, so with a certain level of force, and then it can move one object from one place to the other. So in this case, mechanical work is being done and the load is smaller than the tension. So now let's go back to the diagram. If we begin with a 5 gram as a load, and muscle only need to produce 5 grams of contraction force, and if we look at the lower panel, when the stimulus begins, then the muscle force is building up. When it reached 5 grams, the muscles begin to shorten, and then the weight is being lifted. At the end of the lifting, the force starts to decline. So the top one here is there is a level with 5 grams of contraction force, and during that period we say that the muscle is isotonically contracting. That means with constant force. And in the second column diagram on the top here, when the load is being increased from 5 gram to 10 gram, then the muscle now we look at the bottom panel. The stimulus come, muscle contraction force building up. When it reached 10 gram, the muscle begins to shorten, and that is the plateau here is isotonic contraction. At the end of the contraction, the muscle relaxed, and continues with the third diagram on the top. When the force is introduced, the load is introduced to 15 gram, the muscle can still shorten, and you can see that the shortening period now is less. So the muscle isotonically contracting at 15 grams. At the end, the muscle relax. The fourth diagram shows that during isometric contraction, that is, if now the load is 20 grams, is actually bigger than the maximum contraction force is 17 grams. When the muscle produces contraction force up to 17 grams, the muscle can no longer produce even bigger force. That's why this muscle is not able to lift the 20 grams weight. So we say that now the muscle is contracting isometrically. Isometric means constant length, so the muscle is not shortening. Isotonic means constant force. So constant length means the muscle is the muscle is contracting, but the length of the muscle is not changing. Another type of contraction that we are looking at is concentric and eccentric. So this is both involving the changing in the muscle length during contraction. Concentric contraction means the muscle contracts and shortens. That is the same example as what we see in the isotonic contraction. So the isotonic contraction where lifting the weight is a type of concentric contraction. The other type is eccentric contraction. That is, the muscle that generates force, but the length is being changing, that it's been elongated, so it lengthens, and this one actually absorbs energy. This it puts the body in the position that it can contract concentrically. For example, if we look at the quadriceps muscles in squatting, it is with concentric contraction. So if we look at this diagram, we have muscle tendon on the top and muscle as a body in the middle. And during isometric contraction, the muscle length does not change. And during concentric contraction, the muscle is being shortened. You can see that the length is shortened, but the volume is the same. And during eccentric contraction, the muscle is being elongated. So you need to be very careful not to damage the muscle during eccentric contraction, because that is a type of contraction that can easily damage the muscle. So that is why people say that climbing up the mountain is easier than climbing down the mountain. Many injury actually occurred when you're walking downstairs instead of walking upstairs, because it's more eccentric contraction is happening during walking downstairs. So you need to be careful when you're walking downstairs and climbing down the mountain. So far we've talked about muscle as a force generator, as a contractile machinery. Not only that muscle can contract, muscle also have receptors, because the body, the central nervous system, needs to know how muscle is being contracting. It needs to get feedback from the muscle, then it can regulate, it can adjust the muscle movement. So there are two types of muscle receptors that we need to talk about. So these receptors are for coordinated activities, also for the brain to coordinate and to regulate the muscle movement and contraction. The first one is muscle spindle. Muscle spindle is located within the muscles, and it's for stretch receptor. It detects the muscle length. So if the muscle is being overstretched, the signal can be passed from the muscle spindles. So inside the muscle we have muscle spindles, and this signal can be passed by the nerve fibers. So this is a type 1A efferent nerve, which is passing from the muscle towards the central nervous system. Then the central nervous system would be able to detect the length of the muscle. If it's been overstretched, then it would stop being stretched. So that is very helpful, especially during the eccentric movement. Another type of receptor is called Golgi tendon organs. This receptor is located in the muscle tendon, and that is specifically detecting the muscle tension, which is how much force the muscle is producing. And that is also to protect muscle. This one is protecting muscle from excessive activity. So if the muscle is producing too much force, this signal can be fed back to the central nervous system. So here on this diagram, it shows the Golgi tendon organ. So at the bottom here is the bone, and the muscle inserts the tendon to the bone. So within the tendon you have the Golgi tendon organ, and that is a receptor for detecting how much force the muscle is producing, and the signal is being sending by the type 1B efferent neurons back to the central nervous system. So these receptors are very important for coordination of muscle activity. Now, what about muscle adaptation to use? What are the purpose of training, and what would happen if the muscle hasn't been used for a long time? So with skeletal muscle, they adapt to use. So the first one is there is no cell division. So skeletal muscle cannot be divided into new skeletal muscle cells. That is because development-wise, skeletal muscle cells are fused cells, so it has multiple nucleus along the surface. It cannot be going through the cell division and produce new skeletal muscle again. That is why the injured skeletal muscle can only be compensated by stem cells, not by the skeletal muscle itself. When you do training, mainly it changes the muscle size due to the changing of individual cells. It's very hard to increase the number of skeletal muscle cells. So in other words, you can produce or reduce the amount of myofibrils, amount of actin and myosin proteins, but it's very hard to increase the muscle, the number of muscle fibers, the muscle cells. And if you don't use the muscle for a long time, for example, if someone has been inactive, or it's been aging, or due to injury that they have to be better bound, then the muscle can cause atrophy. That is a disuse atrophy. The disuse atrophy is due to the lack of usage. The muscle fibers reduces their size, lose the number of proteins in the muscle, so the muscle is reducing size, and that is in the aging people. You can see that they are getting thinner because they are being less physically active. Their muscle is being not used, so that is a disuse atrophy. And if the nerve supply to the muscle has been cut, the muscle can also cause atrophy, that is a shrinking in the size. That is called denervation atrophy. And what about hypertrophy? If you train the individual athlete, then their muscle size can be increased during weightlifting, and so that is called hypertrophy. Hypertrophy increases the size of individual muscles, but not the number of muscle cells. So the muscle is adapting themselves to the usage. The next question I would like to talk about is linked with a practical in semester B. That is, having talked about the theory of muscle physiology, how do we study muscle function? How do we study muscle physiology? A very important tool that we use to study muscle function is called electromyogram, that is EMG. You're all familiar with the ECG, and you also have EEG, which is the recording of the brain waves. So these are the common tools that we use to study the human function. So what is EMG? EMG is the recording of collective action potentials from skeletal muscle, because ECG is the collective action potentials from cardiac muscle. So that is the difference between EMG and ECG. So the recording of the action potential collectively is electrical signal that you can be recording from the skin. It's a non-invasive tool. In the practical in semester B, we have two parts. Part 1 is the voluntary isometric contraction, that is using a hand gripping. You can come across here is isometric contraction, that is because the muscle is not shortened, and we are using hand gripping. Part 2 is a dynamic muscle dynamic contraction, that is during cycling. So we will talk to you about how we do it briefly, and you would learn the details in the practical. So during hand gripping, this is a dynamometer with a hand, and then you can see the pointer pointing to how much force this gripping can produce. And so because the muscle that is used for the hand gripping is flexor digitorum superficialis, that muscle do not shorten during the hand gripping because this load is actually bigger than the movement. So we're resting the arm on the table and then using the dynamometer to measure the force that this hand gripping can generate. So let's look at the example of EMG signal. So this is EMG recording from the flexor digitorum superficialis muscle. The top row is the EMG raw data. That is the collection of action potential. You can see that you have positive direction. You also have negative direction. That is depends on the recording of the action potential, can be positive and can be negative. So the activities goes both ways, depends on their relative position to the electrodes. The bottom one is the calculated results, because the both negative and positive signal, it's very hard to compare the amplitude of them, because if you say the sum of it, then the positive signal and negative signal can be cancelled with each other. If we wanted to quantitatively analyze how big the muscle is producing the force, we have to use a method that can analyze the results more precisely and see the amplitude. So that is the bottom row here is called RMS of the EMG. RMS stands for root mean squared, which converted the both positive and negative action potential collection with the raw data into positive only. That is because RMS making the individual data squared first, that is to roll out all the negative ones, and then take the mean value of that squared value, then do the square root of the mean value. So when everything is turned into positive, then we can measure how big the EMG RMS signal is. If we make a stronger contraction, then the RMS signal would become bigger, so we can quantitatively analyze the amount of force that can be produced from a skeletal muscle. What about cycling? In part 2, we do cycling, and the main muscles that are involved in cycling are quite a big group of them. The first one is for extension of the thigh, it's the gluteus maximus muscle. For extension of the knee is the quadriceps, that is up front in the leg. And for flexation of the knee, it's the hamstrings group, which is at the back on the up leg. And you also have calf muscles, which is in the lower leg. The calf muscles with gastrocnemius is flexation of the ankle and the knees, which is to assist. And then you also have soleus muscle, is flexation of foot and ankle. So these are all the muscles involved in cycling. And in this practical, we would like to only concentrate on two groups of muscle. That is the highlighted one, the quadriceps and the hamstrings. So let's look at the muscle itself. On the left side is the illustration of the position of the quadriceps muscle at the anterior muscle at the top upper leg, and the right side shows the details of the muscle groups. So these quadriceps femoris, including these three muscles, which is the rectus femoris, vastus femoris, and vastus medialis. So these are the three muscles within that group that we would like to study. And the hamstrings group is for flexation of the knees. The left side illustration the position in the leg, which is on the back up leg. The right side looks at the hamstrings group with different muscles. So you have a long head and a short head for the biceps femoris. You also have semitendinous and semimembranous muscles. So we would like to place electrodes on top of those muscles on the skin. Then we can record the activity from these muscles. Let's look at some examples. These trays look at the raw data of EMG. The green ones are from the quadriceps and the pink ones are from the hamstrings. So at the bottom here you have the RMS calculated amplitude of the EMG. So this diagram shows during the normal cycling that what most people would do. But on the right side is if you're using a pull and push technique. So when you're not only using the push technique, but as one leg was push, the other leg was also pulling, and you can see that the pink activity is being increased. That is because the hamstrings muscle now works harder. And if we look at the one cycle of the movement, we can see that on the left side there is less muscle activity from the pink, which is hamstring muscle, within that time scale. On the right side, you would see more muscle movement within that time scale. That is because the push and pull technique makes more use of the hamstring muscles. The experienced cyclist would have the technique of push and pull. So because this would increase the speed of cycling, make better use of the muscle groups. So that's the end of the muscle physiology lectures. I hope that you get the muscle function and characteristics, and you also grasped the structure of a skeletal muscle. You can explain the neuromuscular junction and excitation contraction coupling, and you would also be able to describe the factors that contributing to contractile force, and also the different types of muscle contraction and muscle receptors. And you will also have a basic theory towards the practical in semester B. Thank you.